S. Corey. I am a Bachelor of Science in Biology undergraduate student in Jacksonville State University, and my professor is Dr. Benji Blair. My project is about an increased growth of rice in hydroponic culture with the addition of Pseudomonas aeruginosa to the growth culture. And my presentation is the agenda of my presentation is a statement of the problem, project overview, research, variables, hypothesis, cost of the materials, the methods, conclusion, and references. Hydroponic is a proven method to grow rice in Japan. And hydroponic is an alternative method to growing plants without soil using sun, gravel, or liquid with added nutrients. Can Pseudomonas aeruginosa be beneficial to the rice plant's growth? We haven't tested yet, but with the possibilities of increased nitrogen exchange and increased chlorophyll, the possibilities could result in positive aspects. Nitrogen is a major component of chlorophyll, and chlorophyll is act as a catalyst for photosynthesis, which chlorophyll is a vital for the photosynthesis. The green substance in produces the traps light energy from the sun, which is then used to combine carbon dioxide and water into sugars in the process of photosynthesis. And chlorophyll is vital for photosynthesis. Again, the major component of chlorophyll is nitrogen. The project overview is to add the bacteria to rice plants at different stages of growth, seed germination to young growth plant, and expose plants to bacteria after 30 days, 60 days, 90 days, 220 days. I will grow the rice in 30 days increments, starting with young plants and plants grown in 30 increments to see the effects of 1 microliter of Pseudomonas aeruginosa per 100 ml of hydroponic medium. The rice control and test growth will be measured for differences. This research is to identify if the bacteria Pseudomonas aeruginosa has a beneficial impact to the rice, the growth of rice, to determine if the rice will grow through this type of method, hydroponic method, with the treatment of Pseudomonas aeruginosa, and to have an easy ways of collecting the physical data of rice growth by using this type of method with exposure to Pseudomonas aeruginosa. Variables. I have the control variables, the independent variable, and dependent variable. The control variables are the hydroponic medium, the temperatures. The independent is the Pseudomonas aeruginosa. Independent variables is the color of the leaf, length of the leaf, and the roots. It will be color of the leaf, length, and roots will be measured. Procedure. This is an anticipated type of pictures. This is not the real pictures because I didn't test it yet. But my experiment will be almost like this. The rice seeds will be grow on a hydroponic tray. It will be filled with hydroponic medium for germination. Then it will last for 10 days. After 10 days, it will grow like a seed, we call it seed leaf seedlings, and it will be transferred to the test tubes. And these test tubes will be filled with hydroponic medium and treated with Pseudomonas aeruginosa, 1 microliter per 100 ml. So I'm going to make like 12 test tube as a trials and 12 test tube as a controls. The next stage, I will grow the plants and treated them will grow simultaneously three different rice growth: the vegetative stage, the reproductive stage, and the ripening stage. So there will be like 6 aquarium container. I use the aquarium container for the purpose I can observe the growth of the roots. So that one equivalent of approximately 29 to 30 gallons of aquarium container and covered with the styrofoam, hydroponic styrofoam. Then there will be like a hole, like 6 in one row. So there will be like 12 trials, 12 holes, and plants, rice plant bundled will be grown in that styrofoam, in that container, and it will have like 12 trials treated with Pseudomonas aeruginosa solution and 12 trials as a control. So for the 3 stages, it will be a total of 6 aquarium container. On the right side, there's like a typical picture of a healthy normal leaf roots in the green leaf color of rice plant. This is an anticipated results of the growth plant group. The y-axis is like this, measured the centimeters of the plant growth, and then the x-axis like the length of the roots, the length of the leaves, the height of the shoot, and the circumference of the leaf. It will be measured by the use of ruler, of course. So the result will be no effect, the plants will die, or it will weak, or it will become stronger plant. There will be a ripening phase of rice growth, any change in color or taste for use, desirable or not, that will be the anticipated result of the experiment. But expect the younger plants to die, may see some possible change in leaf color to be darker or green, the size increase of nitrogen, the possible change in roots, of size of leaves. That will be all the anticipated result of the experiment. The data from all of the 6 controls can be provided and average then make comparison with the test data on the another tested plants. The observation of the experiment growth with the addition of the Pseudomonas aeruginosa takes about 30 to 60 days, higher or lower survival rate. Cultures with bacterial treatment plus the controls for all these 3 stages, or you call it phases, like the vegetative phase, the productive phase, ripening phase, were tested all at the same period simultaneously. And if this project will have a positive results, it will be a big help to the economy. Why there will be less water is needed to bring the rice plant to the table. I mean, the rice plant is to maturity. Materials that used to be in this experiment. I have the budget of 2,402.48, excluding the electricity cost, the disposal of the waste materials. It will be to be determined. But if this project will be granted, it will be grown in the Jacksonville State University greenhouse. Then the expenses will be lesser. So these are the references that I use to research this kind of experiment. Thank you for listening.